Okay, so welcome everybody to the second session of RECOMB-Seq that is going to be focused on cancer genomics. So before starting with the presentations, let me recall the format. So we're going to have 3 presentations, and each will be of a total of 20 minutes. And we're going to leave 2 to 3 minutes for questions at the end of each slot. And I ask you please to ask questions using the Q&A panel, so I will be able to read them to the speakers. Okay, so I guess we can start with the first presentation that is going to be given by Ziwei Chen from the University of Chinese Academy of Sciences, that will talk about BiTSC2, Bayesian inference of tumor clonal tree by joint analysis of single-cell SNV and CNA data. So please, Ziwei, you can go.
Thanks for the organizers. Hi, my name is Ziwei Chen, and I'm a PhD candidate from Chinese Academy of Sciences. Today, I'm honored to share with you our recent work, BiTSC2, Bayesian inference of tumor clonal tree by joint analysis of single-cell SNV and CNA data. In this presentation, I'll give a brief review to background and related work. Next, I show you our proposed models BiTSC2 in detail. Then I display some experimental results. Cancer can be viewed as an evolutionary process, during which somatic mutations originate from single cells and accumulate in cell populations. Different cells in the tumor may acquire different somatic mutations, resulting in heterogeneous tumor. The cells or populations in the same mutation genotype is called a clone or subclone. Characterizing intra-tumor heterogeneity and understanding the evolutionary mechanism of cancer are believed to guide the principles in predicting and controlling tumor progression, metastasis, and therapeutic responses. Thus, identification of clone or cell genotypes and reconstruction of their evolutionary tree becomes the most crucial questions in the study of tumor evolution. Somatic mutations in forms of single nucleotide variation and copy number alteration, or in short SNV and CNA, are two of the most important sources of information for tumor evolution inference. The rapid advances in single-cell sequencing technology have greatly enhanced the resolution of tumor cell profiles. The SNVs and CNAs extracted from single tumor cells are expected to better characterize intra-tumor heterogeneity. In recent years, great efforts have been devoted to develop computational methods for inferring tumor evolutionary tree by utilizing sources from either single-cell SNV or single-cell CNA. To list a few, methods built on SNVs are mostly maximum likelihood or Bayesian-based model methods. Some of these methods make explicit use of infinite site assumption. For SNVs, there are also a few methods proposed for CNA detection and evolutionary history inference. Recently, we proposed a robust clone or model-free algorithm based on the low-rank matrix decomposition method to recover clone genotypes and reconstruct the clone tree. Robust clone can be applied to raw cell genotype matrix from either source of single-cell SNVs or CNAs. It is accurate in genotype recovery and is efficient and scalable to large-scale datasets. For example, we applied the robust clone to single-cell CNA datasets of breast cancer from 10x Genomics, which contains more than 9,000 cells and 50,000 beans. Robust clone can effectively reveal the spatial progression pattern in the tumor. Nevertheless, is it sufficient for tumor evolutionary inference with only one source of marker? In fact, Both SNV and CNA markers play an important role in tumor generation, progression, and metastasis, and they constitute the crucial traits in characterizing intra-tumor heterogeneity. Notional inference with only one type of marker may lead to biased estimate. For example, suppose there is a true notional process with 5 subclones. If one infers the tumor clone tree with only SNV marker, then a linear evolutionary history tends to be recovered. However, this is biased as it misses the identification of extra subclone 3, which is generated by a copy loss at locus A. It also ignores the CNA driving loss of SNV at locus B in subclone 4, which leads to the misplacement of cells in subclone 4 as the ancestor of cells in subclone 2 and 5. On the other hand, if one infers a tumor clone tree with only CNA marker, then a modification evolutionary tree will be obtained, where cells in subclone 2 with SNV at locus C and B will be mistakenly inferred as the ancestor of cells in subclone 3. Therefore, it is important and necessary to jointly apply both SNV and CNA markers for tumor evolutionary inference. To the best of our knowledge, the only method for tumor tree inference from single-cell DNA data that integrates SNV and CNA information is SCARLET, which optimizes for a loss-supported phylogeny. SCARLET adopts a two-step strategy. First, it constructs a copy number tree with the existing method. Then, refine such tree with SNV profiles of the observed cells and loss-supported cells. However, SCARLET may fall into local optima with integer linear programming of and the subtree root may be misplayed during the refinement. For example, in the above toy model, SCARLET ignores the CNA driving loss of SNV at locus B and make misplacement of cells in subclone 4 as the simplings of cells in subclone 2, which doesn't reconstruct the most parsimonious loss-supported phylogeny as shown in ground truth. Since the importance of jointly modeling SNVs and CNAs, here we propose the BiTSC2 algorithm, which is a unified framework by generalizing sci-fi model to perform Bayesian inference of tumor clone tree by joint analysis of single-cell SNV and CNA. BiTSC2 takes the observed total rates and mutant rates of single cells as input, and models them by a tree-coupled generating model that integrates both SNV and CNA mutations. The model is developed under a Bayesian framework, which also accounts for single-cell sequencing errors. By applying MCMC sampling, we can simultaneously estimate the subclone assignment of cells, the tumor subclone evolutionary tree, the subclone SNV and CNA genotypes, and more specifically, the overlapping relationship of SNV and CNA. In the tree-coupled generating model, we coupled the copy number profile L and the point mutation profile Z by the clone tree T, and model the generation of total race D and the mutant race X under over-dispersion. More creatively, we introduce here a phase indicator G to reflect the interactions between overlapping SNVs and CNAs. We consider 3 possible scenarios of relationship. Firstly, SNV and CNA occurred on parallel branches. Secondly, CNA happened before SNV. Thirdly, CNA happened after SNV. For the first 2 scenarios, CNA doesn't alter the state of SNV. In the third scenario, however, if CNA is a mutant allele, it will alter the state of SNV. The phase indicator G is proposed to comprehensively describe this state change under the third scenario, whereas G equals 1 indicates CNA happened after SNV and affected the mutant allele, and G equals 0 otherwise. Without this indicator, the loss process of SCARLET may confuse with the parallel case of SNV and CNA in scenario 1 with the sequential case in scenario 3. In addition, it cannot detect the gains of mutant copies from the data. As an example, let's recall the previous toy model. We apply BiTSC2 on toy model where SCARLET doesn't recover the most parsimonious loss spotted phylogeny. BiTSC2 can accurately recover clonal tree structure, SNV and CNA genotype matrix, and the phylogenetic relationship T between SNV and CNA where there exists CNA driving loss of SNV and locus B. In order to infer the unknown parameters in the complex space model, BiTSC2 adopts MCMC algorithm. We have employed various sampling strategy with the majority of parameters updated using MH sampling. The subclone assigned C and SNV are CNA origin matrix Z0 and L0 are sampled by Gibbs smooth. For clonal T, we apply a mixed strategy where MH sampling and slice sampling are jointly adopted. Under the MH move, the new tree is proposed by simply random through a leaf clone and we attach it to a random parent node. We treat the phase indicator with net variable when sampling other parameters. They estimate by maximizing their marginal probability after each sampling step. Finally, the best fit number of subclone is selected by a modified Bayesian information criterion. We systematically compare the performance of BiTSC2 to some mainstream algorithms under various simulated scenarios. First, we consider the scenario simulated with CNA driving loss and gain of SNVs. The simulated ground truth includes events of CNA driving loss of SNV happened in subclone 4, and the events of CNV driving gain of SNV happened in subclone 5. Those are the corresponding CNA and SNV genotype matrix. We repeat the 10 datasets under this setting. We compare BiTSC2 with 3 recent works, 3 recent algorithms from results. BiTSC2 can powerfully model the phylogenetic relationships between SNV and CNA, where BiTSC2 accurately estimates the overlapping relationship of SNV and CNA on 7 out of 10 datasets, and completely identified the CNA driving loss of SNV on all datasets. However, SCARLET only models CNA driving loss of SNV, but cannot handle CNA driving gain of SNV. Then BiTSC2 outperforms the other algorithms in tree reconstruction as measured by MP3 distance in SNV genotype recovery as characterized by average, and in subclone assignment as assessed by ARI. Then we consider the scenario where CNAs don't alter the clone structure. Under this scenario, the tumor evolutionary tree can be reconstructed only with SNV marker. We simulated 150 datasets under different parameter settings, which are divided into 5 groups, denoted as G1 to G5. We compare BiTSC2 to the clone and the beam, the 2 algorithms based on SNV data. Compared to SNV-based methods, BiTSC2 recovers genotype matrix with high accuracy and robustness, even at low depth and high missing rate. The error rate of SNV genotype recovery is close to 0, The accuracy of robust clone are being significantly improved as the sequencing depth and the number of locus increase. For subclone assignment results, BiTSC2 and robust clone are mostly consistent with the ground truth. Beam is slightly less consistent. For tree reconstruction accuracy, with measured in MP3 similarity, are almost over 0.9 in all cases for BiTSC2. For robust clone, the tree reconstruction are also good in simulations with moderate missing rate and fewer subclones. Beam is very sensitive to sequencing depth and the number of locus. We also test the BiTSC2 on real SNV data of colorectal cancer patients, which includes both primary and metastatic samples with 182 cells and 36 locus. In the original paper, they inferred a mutation tree with metastasis by polyclonal seeding. SCARLET also often analyzed this dataset, but they inferred a tree with monoclonal seeding event. In our results, metastatic cells are mainly concentrated in subclone 7 and 8, which indicates that the liver metastasis from colon is a single event, which supports the monoclonal seeding hypothesis and is consistent with the inference based on the SCARLET tree. Let's recap the BiTSC2 algorithm. BiTSC2 uses a tree-coupled generating model to integrate SNV and CNA and performs the Bayesian inference of tumor clone tree. It accounts for sequencing errors and is able to estimate the missing rate and the dropout rate. BiTSC2 shows high accuracy in genotype recovery and subclone assignment. This is robust in dealing with SNV data with low sequencing depth and variant missing rate. And very grateful to my collaborators. Now welcome to questions.
Okay, thank you, Ziwei. So we have a question from an anonymous attendee. So the question is, do subclones observed in the data only are arranged into the final tree, and are unobserved common ancestors also being reconstructed?
In my algorithm, the observed common ancestors will be reconstructed on the clone tree. Yes, we can reconstruct it.
Okay, thank you. So I see another question. Maybe I can ask one very quick. So given that your method is Bayesian, did you experimented with different priors on the parameters, and also did you observe a lot of variability among the solutions that you find at the end of the iterations of your algorithm? Okay, so maybe that wasn't clear. So maybe I can ask another from another attendee. So how do you compare it with ISM, and how you generate the CN tree?
We consider the mutation only happened only once on the locus, but we can recover the CNA driving gain of SNV. The CNA tree is we can reconstruct the tree is a jointly tree of SNV and CNA is not a CNA tree.
Okay, thank you very much. So I guess we can move